Welcome to the Neuroccino. As you know, the Neuroccino is a weekly online journal club. And to take note, we have moved the timing from 10 a.m. on Monday morning to 9:30 every Monday. And the idea is you bring a paper, you grab coffee, and you join us for a little discussion about last week in science or historical papers as well. If there's any papers of note that you want the community to know, you're welcome to bring historical papers as well. That's it, basically. That's the rules in terms of asking questions. If you don't understand something, chances are other people don't understand it. So there is no wrong questions. Just ask and we see if we can answer the questions all together. Now with that, we have 4 papers today, I think. So I give the floor to Michel, who's going to start us off. Over to you.
Good morning. Can you all see the paper?
No.
Yeah, no.
It's loaded. It's okay. Yep.
Yes, so we can see it.
All right. Superficial white matter imaging, contrast mechanism, and whole brain in vivo mapping. So this paper dragged my attention. Yeah.
No, sorry.
This paper really dragged my attention because, as you may know, there is a huge gap between neuroimaging and true biology. And in this paper, the authors managed to really bridge the gap between those two using iron imaging. So that's like T2 star. And made is that superficial white matter got a very high content in iron. So they did that in postmodern imaging, and they did these beautiful images here that you can see, where you have the gray matter that has been highlighted in yellow here, and right next to the gray matter, you see those U-shaped fibers that show a high signal over here, which is supposedly with neuroimaging related to the content of iron, okay? And so like they moved on to looking at things closer and checking whether this was really related with iron by de-ironing the tissue. So they scanned twice the brain in its normal state, and after removing the iron using, you know, a chemical approach that I don't know about. You can see how the hypersignals that you see for the U-shaped fibers disappear once you remove the iron, demonstrating that really the neuroimaging approach that they have is able to pinpoint the intensity in iron, okay? And then they went further, they were like, okay, but is it related to the iron inside the myelin, or is it related to outside the myelin? So they actually stain the myelin and look at the relationship with the distribution in iron, and you see that you don't have a straightforward relationship, but it kind of look like this because you got a hyperintensity on the border here, and you don't have really hyperintensity over here for the myelin. So this is not entirely due to the myelin itself. But we know that there is definitely a link with the iron. And then they moved on, and they've been able to show that actually the real relationship is with oligodendrocyte. As you may know, oligodendrocyte are the cells that are connected to the myelin and feed the myelin sheath. And they also contain a lot of iron. So there is a link between U-shaped fibers and high myelin content, and this increases the amount of iron that can be detectable with neuroimaging. And then they did those maps that are showing you the level of iron and the areas that should be then more connected by U-shaped fibers, okay? So the discussion is all about the validation and the interest of the method, but like the real advantage with that is now you can use this sequence to really quantify iron and the myelin in the U-shaped fibers and derive from this information change in the phenotype or like the relationship with disease by comparing these images between patients and controls. Thank you. That would be it for me. Any questions?
So I don't really have a question, but I guess points of discussion. So personally, I find it really fascinating that we can study iron in the brain. Do you know how complicated it is to do it, how reliable it is to compare it to myelin, and if there has been any studies on iron deficiency? If you're iron deficient, you have less iron in your brain, and what does that mean?
So how complicated it is, I don't know, because I didn't try to set up the sequence. I discovered this paper that was published last week, and I think we should investigate it because it's linked with white matter, and we are white matter obsessed people. Then the relationship between the iron level and people with iron deficiency, I don't know, it's been ever reported or explored for the brain. I mean, it's a good question. Never thought about that, but
I guess it would be the kind of the same problem with the intensity in general. I don't know if it calculates a really accurate value of iron. Maybe you would have to compare iron deficient populations to, so you would just have a ratio.
It looks pretty accurate if you compare A and B.
Yeah, but you can. I don't know if you can say like if you take the same image of the same person, will you have the exact same values?
Usually, you use a region as a reference, like like a ROI, the cerebellum, or like the central, like in pathologies, the regions that you know that is not alterated, to estimate the iron content.
But does that mean you need to have a region that is very low in iron?
No, a reference that you know is you suppose is stable. Usually, like in the clinical practice, they do like this. They know that that region should not be affected, and they use as a reference to evaluate at a single subject level the iron content. Usually.
So the part that has the highest iron is the red nucleus, isn't it?
As far as we know, and that's why it's red in postmortem sections, because it rusts literally. So one thing that I learned when I discussed with Carziles about like staining is that he said like staining is very good to see where you have things, but very hard to use for quantifying how much you have. So it's the same problem. The longer you stain a tissue, the brighter is going to be the contrast. And then, you cannot see if that was a lot or a little. It's very variable from brain to brain or region to region according to the diffusion of your staining.
So hang on, staining is just like staining fabric. The longer you leave it in, the brighter the color.
That's exactly what it is.
Interesting. So how reliable then, especially if that is the case, how reliable then is the comparative measure with myelin, for example?
What do you mean?
As in, if you measure the iron with the MRI scanner,
oh yeah,
you get one value. It doesn't matter how long you image it, supposedly. But if you then compare it to postmortem staining, for example, to get the true value, that true value is not a fixed value. Right?
Exactly. Yeah, that's a problem if you want to do several subjects. If you would only do one subject, you might have different staining according to the that you use. Okay, so the intensity might not be exactly the same for.
Yes, that's what I was asking. Yeah,
and
I mean that comes back to this problem as well.
But I imagine for MRIs, that's also an issue because you have different way, things can be recorded, did the patient move or not, or these kind of things. Yeah. And we try to correct for it. The great advantage is if you do a correlation, like a Spearman correlation, everything comes back into proportion. So if you do a Spearman correlation for one slice of stained tissue with MRI, then they will be comparable because it's an order relationship. If you do a separate slice, then you start like screwing up your order, and then like your relationship might be lost or smaller than expected.
Okay, interesting. Definitely worth investigating.
Cool. Let's move on to the next paper.
So we had Patrick who had a paper. No? Okay. Leah? Let's come back to Patrick after.
Okay.
There you are.
Leah, do you want to go first?
I am sharing the screen. Okay.
Fantastic.
Can you see it? So this is a study, not about last week. It's 2019 neuroimage, and is about the relationship between age and axonal microstructure in the corpus callosum. They use the connectome scanner by implemented a sequence for diffusion using Eiger gradient, 300 millitesla per meter, and they also use like different lengths of the gradient pools, different b values to evaluate the axonal diameter density of the axons in the corpus callosum. They evaluated first the corpus callosum as a whole, then in the different subregions segmented using free surfer, and they compare these the value of their model that evaluate the axons as a simple cylinder, and they correlated these with also metrics that are currently used in the clinical practice as FA, RD from the DTI model, and also multi-compartment models like kurtosis or NODD. And the population was 36 healthy subjects ranging from 22 years old to 72. They also compared in a subgroup of 10 subjects, like the older one, a correlation with the clinical scores, and in particular they found interesting results about short-term memory, not really significant because the group, the size of the group was like only 10 subjects. Going to the results, they found interesting results, in particular in the genome of the corpus callosum. Going back to the figures that maybe are more clear. So, so this is the division that you have if you use free surfer. They also evaluate cortical atrophy as the gray matter volume divided in the different lobes, and also also subdivision like prefrontal, frontal. And this is an example of the results, like in a young subject and an old subject. You can see the different distribution, in particular here in the genome, in this posterior portion. And if you look at the correlations in the corpus callosum as a whole, you see significant correlations in the axonal diameter index with age, in the axonal density, not in the FA, but in the FA if you consider the whole white matter. Then if you move to subregions, you find significant correlations in the genome, and also in the in this posterior region with age, and this is the axonal diameter. And then if you compare these findings with the DTI metrics, you find a correspondence with the AD, RD, and also FA. So there is an agreement between this high density gradient estimation of the axon density and the DTI matrix that is interesting. And you can find, and also they use an atlas-based approach using the Hopkins atlas to evaluate the forceps minor and major, and they find a correspondence only in the forceps minor with the age. That is like all this correspondence is more in the frontal lobe. And they found also this evaluating the correspondence with the axonal diameter and the gray matter volume. They only find an agreement in the genu and the frontal and the prefrontal cortex. And yes, these are the parameters. So yes, in this paper you can see that there is a correlation with age and DTI, like microstructural measures, both evaluating using Eiger gradients, but also using the conventional DTI models. They don't find an agreement using the Notti and kurtosis models. They found other specific like measures, but not this correspondence. So I found this interesting because I was looking about aging, how our brain change with age, and I found this article. And it's really clear if you want to read it. Like the methods are really clear, but the sequence, of course, is a good sequence. Like in the limits, they say that if they infer the axonal size, they can evaluate a size of 2.6 micrometers, and they know that this is a limit because there are also axons that are like smaller, but I think that it's really nice to see how like the measure that you can have with the Conetom scanner can agree with the measure that you have in with the value of about 100 in the clinical practice. So there is really correspondence, and also it's interesting that something is happened in the frontal lobe, and also they said that if they correlate the axonal diameters with the cortical atrophy in the prefrontal cortex, correcting by the FA, they also have a correspondence. So there is something that is going on there. So I find it interesting. Also, this translation to the most advanced sequences to the currently used. And also, if they evaluate cognitive scores, they found a correspondence with short-term memory. So there was an atrophy and an increase in the axonal diameter in the genu, and the atrophy in the prefrontal cortex. Lower were the scores for short-term memory. So something that is interesting. Any questions?
Thanks, Leah.
Welcome.
I was wondering, the result that struck me the most that you just presented was that axon diameter appears to increase with age.
Yes, so, because the density is lower, but the diameter is higher. So yes, you're right. So you see the density, so it's like they are like less axons, but like yeah, bigger. So it's like the contrary of atrophy. So if you have more space, maybe they are not so packed, and they are like, they didn't discuss this point because they like say yes, it's so they are less, so less density and higher. I don't know if it's something in the model because you estimate the intra-axon diffusion, the extra-axon diffusion. So if, yes, maybe it's something also in the model. But the idea, I think that you have less axons.
But I mean, from a biological perspective, it would be quite intriguing to see that axons would increase their diameter, their volumetric shape as soon as they got more space, right?
Yes, they also say
would,
that these findings are in agreement with some histological studies and postmortem studies. So they were really sure about this. I
really didn't expect that. I would have thought that more of the room that becomes free would be infiltrated by simply corticospinal fluid or something like that.
You see, in a postmortem study of 20 individuals, this is the increase, large callosum fibers consistent increase in the gene of the core in both males and females. They were really sure about these results, and they discussed with histopathological findings.
I think Michel also has a question.
Okay.
Michel,
Yeah.
Okay.
Leah, I got a question for you. So it's very interesting paper, but when you come to the correlations between axonal diameter and axonal density, it looks like the correlation was higher. I think. Oh no, it was okay. It looks like axon density is a mirror of axonal diameter, which kind of made sense in the biological perspective, but I was wondering whether the models that estimate axon density and axon diameter derived from diffusion, whether this model, like the mathematical model, like the two measures can be influenced by each other. You know, you're physicists, so you know what's behind it, but if you put the two equations next to each other, is there like some linearity in the estimate, collinearity in the estimate?
So yes, they actually use, maybe we have to move to, okay, they use a really simple model. There are also more complex model. Their model also only have these three compartments. So you have like, they model the axon as a cylinder, and they evaluate the fraction of the volume that is inside this cylinder outside, and the outside is evaluated as the CSF. So it's a really simple model. So of course you can have, like, I think an inference. There is a correspondence between the two measures, but I think that I prefer this simple model than other models that are more complex. So they are trying they're trying to like evaluate all the possibilities or the interaction between the interaxon diffusivity and extra-axon diffusivity, but then you have to put in the model a lot of parameters that you have to suppose that are like this. So if the model then is complex, you have to you have these parameters that you have to estimate in some way. This is why we use multi-shell data, and so they prefer to use this the simplest model that you can have. So of course there is the correspondence, because if the density is related to the axon. They didn't comment this, but I can look into it. But yes, I think.
I mean, it's just out of, say, curiosity, but you could, you know, because you have like three measurements, you can just plot the three equation into a three-dimensional space and see whether the distribution is biased.
The distribution of the stream.
Of all the values that can come out, because they're interdependent. So you'll see a shape, a 3D shape that will tell you, oh yeah, there is like obviously a strong correlation between the three when it's derived from from this equation. And then maybe you can do the same thing from postmortem. But just an idea, just like.
Yes, they say that they can use also these other models that are more sophisticated, that they combine either a restricted model of water diffusivity. There are other approach, but yes, it's only about the model. They don't project in a different space. I haven't seen that yet.
There's 2 more questions. Chris will first.
Yeah. First, have you linked the paper somewhere? Because I would be interested to read it.
I put
okay,
on
okay.
the Neuroccino website, so you
Cool.
can.
Okay,
find
great.
it.
So yeah, I had a question about the localization of the changes. So like, there are different correlations when you divide the corpus callosum in different parts. I'm just wondering why there is a difference between because most of the axons should be the same, right? I mean, from one part to the other part of the corpus callosum, no?
No, I think that there is a different distribution of the axon population within the corpus callosum.
Yeah.
Yes.
It's just I'm wondering whether if you change the division of the corpus callosum, if you can have another distribution of the correlations. So
yes,
how do they? Is it based on histological atlas, or how they divide the corpus callosum?
So they use the free surfer parcellation that there is MRI underscore CCA function that you can use to divide the corpus callosum in 5 regions. That these are the regions, and I think these regions are, yes, there is like like a geometrical division, but there is also correspondence with the structure of the corpus callosum, if I'm not wrong. So actually, you know that the density of axons is different. The population is distributed in a different way within the corpus callosum. And so they use this segmentation that you have segmenting the T1 image using free surfer. And yes, in their limitations, they say that they can try another segmentation, because yes, you have this bias, this assumption that you made at the beginning.
Yeah.
But
yeah, especially when you don't find the same correlation on bigger parts. Because of course you can always just divide your parts and you will always find a smaller region with a correlation if you want. Of course, if it's based on an atlas, it's a bit like it's less biased, but still you have a division that you chose. Maybe you could have chosen another one and have completely different measures.
Yes, like or a voxel based approach for correlations. Yes, I understand what you mean. Yes, this is one thing that they discussed. Okay. But yes, this is a really common division using free surfer, and it's based on both on geometrical and also there are different population of axons within the corpus callosum.
Okay, thanks.
You're welcome.
To say we're doing perfectly on time. We got 4 papers, 2 done, and we're halfway through. It's fantastic. Let's see, there was one more question from Valentina on this paper, if I'm not mistaken.
Yes.
Yeah, you're back. Welcome.
Hi.
Sorry, I'm not showing the video because I jumped off the train. Did they check in the model, test in the model, the contribution of the education of the participants, or maybe they matched the participants for education?
No, they never discussed this.
Okay.
So now maybe if I don't think also in the method. No, they say that they don't have problems. No, the education was something they do not discuss.
That's a very good point, though, because education has been shown to be relevant. Yeah.
I mean, I was expecting something from education.
No. There is also in the methods part, No, they only exclude like major neurological problems, but they do not discuss. They only evaluate this moral cognitive assessment score, and also in the discussion they don't think that they don't talk about education.
And one last question, but for just naive people, what is a connectome scan?
So usually you see the connectome scan is a scanner that they made. It's in New York, I think, right? So it's a scanner that you change the gradients. For example, this number that is 300 usually in a clinical scanner is about 40. So you can see, and this is what gave you the image resolution. And if you increase this value, then of course, like your coil are like bigger, all the software, the engineering parts is different. Of course, more expensive, but also you have to take care about a lot of things. So the connectome scanner is a scanner that is modified to achieve high resolution, in particular in DTI microstructural measures. And so you have sequence that you can't have in a clinical scanner, because as I said, this value is usually 40. Here you have 300. And this is what changes your image resolution. And so you can have a resolution that is actually microstructural. And so.
As a follow-up question on that, Leah, so that value is determined by the hardware, not the software, right?
Yes, the hardware.
Okay. So you wouldn't be able to implement that on a clinical scanner. You need a different scanner.
Yes.
Okay.
Yes, this is the... yes, it's in the hardware.
Okay. Any more questions on that paper? No. Okay, in that case, we move on to Patrick and his preprint paper.
Hey, hello everybody. Can you see my screen?
Not yet. Here we go. Yeah.
That should have fixed it. So, all right. Last week, my attention was drawn to this preprint that was created by quite a lot of authors and concerns the central questions if the tracks that we're looking at or that we're trying to investigate are somewhat reproducible across groups, because as we know, in order to gain any meaningful insight from our research, we really rely on reliable and reproducible methods. So what the authors here did was that they had a small sample of just 6 participants, but they gave these 6 participants to up to 42 groups with the simple task of dissecting 14 very common white matter samples. And you will see to what extent now the results were comparable across these groups. As I said, 14 different tracks, 6 subjects, 42 groups, and what they received was a total of 57 different segmentation protocols, different ways of how these groups handled processing and tractography of the data. Moreover, they then analyzed to what extent the very different sources of variability existed. As we know, certainly there's variability across subjects, there's variability across the segmentation of protocols, but in fact, the variability across these protocols for the bundle segments was greater than any other sources of variability. If we look at certain tracks, we can actually see that the spatial distribution of these tracks are in fact quite diverse across groups. So for instance, here in this light blue, or yeah, we see the aquatic fasciculus, which always somewhat looks alike, but we see huge variance in the final outcome. Same is true for simple tracts such as the corticospinal tract here. They actually also had a nice map of the distribution of these groups on our planet. So, and also here, it's quite interesting to see that there's a bias. Obviously, when it comes to neuroscience, we do have a cultural bias, so that countries which are richer typically have access to the scanner, have access to the resources, as we only have few, if any, countries outside of the cluster of Europe and America. You also see that different groups decided either to do deterministic tractography or probabilistic tractography. And for each of those tracks that they highlighted, we see this kind of variance. So that's not the best note or best message for our field, obviously. And it really highlights that we need to come together in order to make our research a little bit more reliable or reproducible across groups that are interested in this kind of research. One good note, however, is when they look at, just highlight this really shortly, if you look at the results across dimensions, meaning if all of your tracts are a summary of various high dimensional objects, and we measure this dimensionality, this complexity, and bring it back to 2 dimensions, just in order to visualize how these tracts would have looked like or would have compared to one another in a low dimensional space. This is what is done here using UMAP. And we can see here color-coded that even though across groups there are differences, all these different tracks are somewhat related to one another, so they resemble one another. Hence, we can say that to some extent we all look at the same tracks, but the actual spatial layout and the actual variability that we induce due to our differences in protocols have huge impact on what we're doing here. All right, any questions?
Chris, go ahead.
Yeah, so did they just measure the changes in the protocol? So it's pre-processing and everything to the, I mean, from the raw data to when they dissect the tracts? Or did they also compare just the definition of the tracks, like how they would define a beginning and end points for the tracts?
I need to check that again. I can't really honestly answer this question at the moment. So I think no, got to check it. Sorry for that.
No, it's just because I did they give the images already pre-processed to everyone to give them like one dissection to make with their own protocol and one dissection with a standard protocol decided before, so they can compare that? I mean, that's the first idea I would do to compare the two.
I don't think that they looked at which differences in processing were the biggest. So certainly you can diverse in different aspects of your processing and different at different time points during the actual processing. But I don't think that they have mentioned that.
Okay.
Or probably I didn't I didn't see it.
Yeah, maybe I will read the paper. And,
You got this paper anyway.
yeah. Thanks.
Any other questions? For now? You said they segmented 14 pathways.
Yes.
Why only 14? There's more white matter pathways. Was that all they had in the data, or the tracks that they knew about, or was that a natural limitation they gave?
I think that was the limitation that they gave. So probably these are just the tracks that are most common.
Mm. And then also in figure 1 that you have up on the screen right now, it looks like they compare all the different algorithms. So on the very left, it looks like a classical tensor reconstruction of the arcuate, whereas the next 2 images look more like a spherical convolution algorithm. And that's a difference that's well documented, which doesn't necessarily mean that one is better than the other, but it could mean that depending on the track you're interested in, you need to use a specific algorithm to get the best result, because even though the tensor model is often bashed as the worst ever, it does have its advantages when you come to certain tracks.
I actually don't know if any of the tracks would have benefited from typical or simple tensor-based models, because I really don't think that in this year, like 2020, simple tensor models are preferable to compared to more complex models that actually take into account like crossing fibers, kissing fibers, and anything like that.
It very much depends on what you're interested in. From an anatomical point of view, the tensor model might show you less, but it should reconstruct the core of a pathway. If you're more interested in the lateral projections, for example, then obviously the tensor model will have its limitations. But for the core of a tract, it's still a valuable method. And some pathways, like if you're only interested in the corpus callosum, for example, the midsection, you don't need advanced methods, and not everyone has access to advanced tractography yet. That's the other issue.
Totally agree. I totally agree.
Sure. I find it a bit difficult to just, as a blanket, say tractography based on DTI is no good anymore, because it still has its place, especially in clinical imaging, I think.
It certainly has its place. However, if you can go to a higher, more complex model that takes into account different, like crossing fibers, kissing fibers, these kind of things, then I think, obviously depending on your research question, but if you want, well, I don't know to what extent the reconstruction wouldn't be better if you take a better model, right? I mean, yes, as you said, if you have just the core, if you're just interested in the core, or if you're interested just in the corpus callosum, with no regards of the collaterals of the corpus callosum, sure, you can take the tensor model.
But also the thing is, it's all good to throw fancy algorithms at the data, and you see more, no question about it, because you resolve the crossing, but you're also amplifying your false positives. So rather than just throwing fancy algorithms at it, we should also focus on validating it from an anatomical point of view. Otherwise, we have very nice looking streamlines, but they have no anatomical meaning in the end. So that's one thing to consider. Right, Michel.
Yeah, so like I'm you know, as you may know, I'm really fascinated by UMAP, and if they've been able to embed together tracks according to their anatomy, they should be able to reverse this embedding and automatically dissect prototypical connections from tractograms, one way or another. And I was wondering whether they touch this point in the discussion, or if you think it would be feasible.
they haven't touched it. So the thing is, they didn't even use a clustering algorithm at that point,
Oh, okay.
where they tried to data-drivenly see if those tracks were really distinguishable from a low-dimensional point of view. So I think color 10 here, the upper left, is basically just a depiction where they used prior knowledge to differentiate between the tracks with no classification algorithm. At least I haven't seen that they reported any. In theory, it could be, well, it's difficult to say that it's possible to reconstruct from low dimension to high dimension. Because obviously when you lose dimensionality, you lose information. So you neglect information for the sake of being able to simplify the results. And albeit UMAP captures similarities across different dimensions, it integrates this information about the similarities and then leaves it. So that in the final figure, for instance, here, you couldn't take one point and reconstruct an entire track with it. I doubt that it's possible without having the information about the input matrix, that is.
Fair enough.
Looking at that figure you're just showing, especially in the panel C, it's hard to judge from a 2D image on a shared screen, but it looks like they combined various different tracks in that cluster, including the IFF, the ILF, Meinert's loop, maybe some more. So from an anatomical point of view, that just makes me wonder what we're actually looking at here.
I mean, as I said, they used basically UMAP as a means of just visualizing without doing parametric testing for the UMAP parameters. So it's not like they tried to use the best parameters in order to spread out different tracks. I believe what they did, so I haven't seen a shared code or anything like that, but I believe what they did was to just put the data into the vanilla version of UMAP without further parameter testing. So image C, as you can see here, is also depicted on the left. It just zoomed in version of the panel basically. And yeah, it's totally correct to say that there are quite a lot of tracks here that are actually different fiber bundles. As we can see, all of these tracks share similarities, which is basically why UMAP picked them up as being close together.
So this is one paper in a series of papers that came out over the last few years comparing the different algorithms. So what's the novelty of this paper? Why do we need another one comparing the algorithms?
I'm not aware if any other of the prior papers has done it in such a large scale. You can happily correct me on that, because I don't think I know all of the other papers that compared algorithms. But like, I think 42 groups is quite a number, and also really shows that no two groups basically produce really the same results. And that's kind of frustrating, at least for me, because if we want to understand how structure varies across individuals, or if we want to understand how variability in structure relates to function, relates to behavior, the main source of variability shouldn't be our protocols.
That's why I say we need to come back to anatomy.
Definitely.
All right, any more questions on this paper? No? Okay, then we have one more paper, I believe. Victor, you ready?
Yep, sure. Can you hear me well?
Yep, take it away.
All right. So it's just a small paper that I found that is quite interesting. It's more a fun paper than a groundbreaking paper. So here it is. Can you see it?
Yes.
All right. So it's about the preservation of cerebral tissue in the bodies of people that have died in Herculaneum during the eruption of the Vesuvio 2,000 years ago. So the archaeologists discovered the remains of kind of like imprints of neuron inside of the remains. Basically, it's they say that the neurons have been vitrified by the rapid heating then cooling of the central nervous system. So it seems to be a natural process that allows for the structure to be the hyperstructure of the neurons to be kept. Well, at least some of the neurons you can keep some of the structure of the neurons, and so I thought it was really interesting to find that we can have images. So this is images from just electron microscopy of the pieces they took inside of the brain, and you can see the like dendrites probably and the axons that have been highlighted in green of neurons that are basically 2,000 years old, and we can find in the myelin periodicity of the sheath at different points in the axons. That is quite impressive to see on such an old structures. And they even claim that we can see inside of the structure, we can see the different layers of the myelin sheath. I'm not completely convinced because they do make some bold claims, but it's pretty cool. And so yeah, they also try to confirm that it was really brain tissue and spinal cord tissue by analyzing the proteins that they were surrounded by and doing a comparison with the known database of protein and expressed genes in these areas. So they say that they are pretty sure that these are neurons from the CNS. And so yeah, they claim that this allows the study of ancient brains. Well, not super ancient. 2,000 years old is old, but not. You cannot really study the evolution of humans across this kind of time scale. But still, it's quite impressive that we can see that, and it might open the field for this kind of research in ancient samples, but it requires very specific conditions. So quite rare, but still quite interesting, I think.
Okay, thank you very much. That paper did make the news because it's interesting to history buffs and science buffs alike. And especially amongst my Italian friends, they were quite intrigued by this discovery. Do we have any questions? No? Okay. I'm just really fascinated that they, like as you say, it is old, but it's not super old. But still, it's old enough to anticipate that you wouldn't find a brain cell anymore. So this is quite fascinating, I think.
Yeah, I'm really interested in how exactly this vitrification process works, because I have no idea how you can keep such a fine structure with such a destructive process as like being rained on with burning ash. It's a bit weird.
I find it utterly fascinating. To be honest, I don't know what to do with that information yet.
It's just a fun paper. It's not supposed to be anything else, I think. Well, at least for me.
Well, who knows? Who knows?
Yeah, who knows?
Okay, Michel?
All right, it's a different process, but do you think it would be possible to do the same thing with mammoths trapped in ice?
It's a very different process, yes. With mammoth, thing is, with this kind of process, because it's a very fast process that happened, it was like scorching hot temperature, then it cooled down really fast. This kind of fast process allows for the conservations of fine structures. It's the usual stuff. You need something very fast to keep these fine structures. For mammoth in the ice, I think it's usually it takes a longer time for the whole mammoth to freeze over. So I think it'd be more destructive. But I also, well, I'm no specialist, but I've heard that there is possibility to find neurons in mammoth and stuff like that, because there is more sugar and more fat tissues in well, there is stuff like that in the brain, and it can act as an antifreeze process that keeps some of the neurons more or less intact. So I have to check that out, but I've heard something like that a while ago, and we might have something really in mammoth. I don't exactly know, but it's a completely different process.
Okay.
I have a question.
Go for it.
Do you think that one can predict the kind of neurodegenerative disorders that people had, or I don't know, whether they suffered from stroke or from tumor?
I think it's unlikely, because I don't think they managed to get that many samples, and maybe if you look out and you find like the neuron with the myelin sheath getting degraded over time, and you see that at some point, and you say, oh, this is exactly the same that we have in patients, and you say, yeah, maybe, but you have to be lucky, and you also have to be sure that it's not a degradation that is due to the fact that, I mean, the person died because of very extreme temperatures. So I think it's pretty hard to draw conclusions about the health of the person based on this kind of data.
Thanks. But it's very interesting paper.
Yeah, I think it is.
It's definitely a fun paper. Okay, are there any more questions on this one? No, okay. In that case, I think we discussed all the papers. Before you leave, I would like to just test with you quickly our poll. And for those of you who are co-hosts, you won't be able to answer the poll. And everyone else, I just took away your co-host privileges. So I'm just going to launch the poll now and give you a minute to fill it in. If you could just let me know that you can see the poll. Okay, and if you could try to fill it in, because recently we had an issue with that. So just want to check that it's working. Yes, fantastic. Okay. So I can see 3 out of 6, 4, 5. Fantastic. Okay. 6. Fantastic. Okay. 80% Europe, 20% UK. Background, psychology, 50%. Neuroscience, 67%. Neuroimaging, 67%. Physics, 17%. Biomedical engineering, 17%. That's one person each. And we know who you are. Highest degrees, we have a mix of MSCs, 33%, and 67% PhDs. And how did you hear about today's event? 67% word of mouth, 20% Twitter, and 33% of the website. So I'm just going to quickly show you the results. Nicely highlights the majorities. And with that, let me stop sharing. Here we go. Glad this is working. Well, thank you everyone for coming this week. It's been a pleasure. It worked really well. Just as a reminder, the new time is 9:30 every Monday morning. And spread the word, the more the merrier, and keep bringing fascinating science every Monday. Have a good week. Bye.
Thank you.
Bye.
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 Thank you. Bye.
Bye.